Hello, welcome to Hydron Academy. In today's video, we're going to start the incredible world of microorganisms, specifically, Eubacteria. These microscopic beings may be tiny, but their impact on our planet and daily lives is immense. From helping us digest food to shaping entire ecosystems, Eubacteria are fundamental to life as we know it. By understanding Eubacteria, we gain a deeper appreciation of biology, medicine, and the environment. So, get ready to explore their fascinating structures, functions, and importance. Let's begin our journey into the unseen world of Eubacteria. Eubacteria, commonly known as true bacteria, form one of the most diverse and abundant groups of prokaryotic microorganisms, which lack a nucleus and membrane-bound organelles. Here are some general characteristics of them. They exist almost everywhere, thriving in extreme heat, freezing cold, acidic conditions, and even inside the human body. Whether in soil, water, or living organisms, these microscopic life forms contribute significantly to various biological and ecological processes. Unlike more complex cells, Eubacteria have a simple structure. They lack a nucleus and other membrane-bound organelles, yet they have evolved incredible survival mechanisms. Some bacteria play a beneficial role, such as aiding digestion and nitrogen fixation in plants, while others can cause infections and diseases. Despite their simplicity, Eubacteria are powerful, adaptable, and essential to life on Earth. The structure of a bacterial cell is unique and well adapted for survival in different environments. These cells are usually microscopic, ranging from 0.1 to 5 micrometers in size. Despite their small size, they possess all the necessary components for survival and reproduction. A bacterial cell is enclosed by a cell membrane, which acts as a barrier and regulates the exchange of substances in and out of the cell. Surrounding this membrane is the cell wall, a rigid structure that provides support, protection, and shape. The composition of the cell wall varies among bacteria, influencing their classification and response to antibiotics. Inside the cell, the cytoplasm is a gel-like substance where essential cellular activities take place. Unlike eukaryotic cells, bacteria lack a true nucleus. Instead, their genetic material, DNA, is found in a single circular chromosome that floats freely in the cytoplasm. Additionally, bacteria contain ribosomes, which are responsible for protein synthesis, allowing them to grow and function efficiently. Some bacteria also have additional structures like flagella for movement and pili for attachment to surfaces or other cells. Bacteria come in various shapes, which help scientists classify them into different categories. The three primary shapes are cocci. These bacteria are spherical or round in shape. Examples include Streptococcus, which can cause strep throat, and Staphylococcus, often associated with skin infections. Bacilli, these are rod-shaped bacteria such as Escherichia coli, commonly found in the human intestines where it aids digestion but can sometimes cause infections. Spirilla, these bacteria have a spiral or corkscrew-like shape. A well-known example is Treponema pallidum, the bacterium responsible for syphilis. Each shape plays a role in how the bacteria interact with their surroundings, move, and infect hosts. Some bacteria also form clusters or chains, further influencing their behavior and function. The bacterial cell wall is one of the most critical components of a bacterial cell. It provides structural support, protects the cell from environmental stress, and determines how the bacteria interact with antibiotics. The cell wall is primarily made of peptidoglycan, a mesh-like polymer that gives bacteria their strength and shape. Based on cell wall composition, bacteria are classified into two major categories using a technique called gram staining, gram-positive and gram-negative. Gram-positive bacteria have a thick layer of peptidoglycan, which retains the violet stain during gram staining. Examples include Streptococcus and Staphylococcus. These bacteria are often more susceptible to antibiotics like penicillin. Gram-negative bacteria have a thinner peptidoglycan layer but possess an additional outer membrane. This outer membrane makes them more resistant to certain antibiotics. Examples include Escherichia coli and Salmonella. Understanding bacterial cell walls is crucial in microbiology and medicine, as it helps scientists develop effective antibiotics and treatment strategies. Bacteria have evolved various ways to obtain energy and nutrients, allowing them to thrive in diverse environments. Their nutritional strategies can be classified into two main categories. They are autotrophic and heterotrophic bacteria. Autotrophic bacteria. These bacteria produce their own food using natural energy sources. Autotrophs obtain their carbon from inorganic carbon dioxide. They can be further divided into photoautotrophs. Use sunlight for energy like cyanobacteria, which play a role in oxygen production. Chemoautotrophs. Obtain energy from chemical reactions such as sulfur or iron oxidizing bacteria found in deep-sea hydrothermal vents. Heterotrophic bacteria obtain at least some of their carbon from organic molecules like glucose. These bacteria rely on consuming organic matter for survival. They are also divided into photoheterotrophs, heterotrophs that obtain their energy from sunlight. Chemoheterotrophs harvest energy from organic molecules. These diverse feeding strategies enable bacteria to be key players in nutrient cycles, environmental balance, and even human health. Bacteria reproduce rapidly, allowing their populations to grow and adapt quickly. They primarily reproduce in two ways: asexual reproduction, binary fission. This is the most common method. A single bacterial cell copies its DNA and splits into two identical daughter cells. Under optimal conditions, some bacteria can divide every 20 minutes, leading to exponential population growth. Sexual reproduction, genetic exchange. Although bacteria do not reproduce sexually like higher organisms, they can exchange genetic material through processes such as conjugation. A donor bacterium transfers a small DNA segment, plasmid, to a recipient cell through a pilus. Transformation. Bacteria take up DNA fragments from their environment. Transduction viruses transfer DNA between bacteria. These genetic exchange mechanisms allow bacteria to develop antibiotic resistance and adapt to new environments, making them incredibly resilient organisms. In this video, we explored the fascinating world of Eubacteria, from their structure and shapes to their cell walls, nutrition, and reproduction. Though small, these microorganisms are incredibly diverse and essential to life on Earth. Some bacteria support ecosystems by recycling nutrients, while others help humans digest food and produce medicine. However, some can also cause diseases, highlighting the need for ongoing research and medical advancements. Thank you for joining us on this journey into the microscopic world of bacteria. If you found this video informative, don't forget to like, share, and subscribe for more engaging content. See you in the next video.